Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome to the first Base to Base biotech podcast, which is kind of a play on face to face. And also, bases are important in biology, and baseball too, I guess. It is going out on March the 21st, so for some people, they're just getting back from Bio Europe Spring in Milan. For those who don't know me, I started my career on the radio in Canada and have since been in the media for a long time, maybe too long, about 30 years. And I was editor of a biotech publication and presented a very successful podcast on biotech for about 3 years for another platform. And I've been encouraged by quite a lot of people in the industry to keep going. So here we are. I think they were saying keep going, not go away. Actually, in addition to today's guests, we do have 9 other interviews already scheduled, so I guess it must be okay. You can find this podcast on one of our websites, deeptechdigest.com, but you can also subscribe to it on Spotify, Apple, Google, and all good podcast platforms, and probably a few not so good ones. It's also on YouTube. I added the word biotech into the podcast name simply for search purposes, and also so people don't think it's a baseball podcast, much as I love baseball. We'll be featuring anywhere from 1 to 3 interviews each week about some of the latest news, whether it's a new drug, a clinical trial, new discoveries, new technology, and of course you can reach out to suggest guest interviews by emailing jim@deeptechdigest.com. There are no commas or hyphens in there. Today we have 2 interviews with Laura Yecies, CEO of Osteoboost, a company about to launch a wearable device to tackle osteopenia. But first, we have a conversation with Roy Mauti, co-founder and CEO of Pheast Therapeutics, which is developing an anti-CD24 antibody drug candidate that is designed to block a key macrophage "don't eat me" signal on cancer cells. Okay, so I guess the first and most obvious and easiest question then is if you could give me a bit of background on the company.
Certainly. So Pheast Therapeutics is a San Francisco Bay Area based company focused on cancer immunotherapy, and we're started based on some science developed at Stanford by two of my co-founders, Irv Weissman and Amira Barkal. We're about 35 people at the moment, so still relatively small, but growing. And as we approach the clinical stage, we're looking to expand not only on the research side, but now on the clinical development side as well. We have a real focus scientifically on macrophage-based immunotherapy.
And I know that you're looking at cancers that are really hard to treat. With respect to that, what are the challenges that you and others in the same field are facing?
Pheast scientifically is really focused on macrophages, and that is for good reason. These are, immune cell types that are extremely abundant in many cancers, including those difficult to treat cancers that you mentioned we're focused on. They often can be counterproductive in the tumor microenvironment. They promote tumor growth. They're immunosuppressive to other aspects of the immune system and can just basically be a barrier to efficacy of existing therapies. So I think for a long time, the immunotherapy field has been looking for ways to effectively pull them into the fight against cancer. And we see that as particularly impactful in some of these cancers that we're currently focused on because you have, unlike the cancers that are well-validated places for T-cell checkpoints to be effective, many cancers lack the initial conditions to be good candidates for T-cell immunotherapy. And so, for example, ovarian cancer is an area that T-cell checkpoint therapies haven't really made a big impact as they have elsewhere. So we think that those indications are really in need of immunotherapy solutions, which, if used properly, can really complement a lot of standard of care paradigms like chemotherapy, radiation, or ADCs, which are ever further impactful class. And so we're really focused on trying to meet this need by harnessing the science, by bringing these macrophages into the fight. And we think it's just a matter of finding the right targets and the right ways to activate them.
I wonder if you could talk a little bit about CD24 and the relevance of that and how ubiquitous that is in the field of cancer.
Yeah, very happy to do so. I mentioned the company was started based on science developed at Stanford by 2 of my co-founders. So that group is Irv Weissman, who's a longtime stem cell researcher, really one of the kind of fathers of the stem cell field. And he and his lab have often had, over the years, had a focus on harnessing the immune system and using it for therapeutic purposes, including in cancer immunotherapy. So the rest of the co-founders, Ravi Majeti, who's the current head of the Stem Cell Institute at Stanford, Mira Barkal, who was kind of the scientist who identified the CD24, the target of our lead program, and myself, all trained at Irv's lab at different times as graduate students or postdocs. So we share this sort of scientific passion and focus. And over the years, each one of us has been really looking to find ways to block the signals that macrophages see in the tumor microenvironment that cause them to be bad actors. And CD24, based on the work that Amira and Irv did, is clearly one of those signals. It's an immunosuppressive receptor, a small unstructured peptide that's really heavily glycosylated, so a little bit structurally different from some other targets of cancer immunotherapy drugs. And it has a variety of functions in the immune system. Going back to the 90s, there are academic papers that characterize a role for it in B-cell maturation or T-cell homeostasis, damage-induced immune responses. And then with Irv and Amira's work, we now have a clear understanding that it is a strong immunosuppressive signal for macrophages as well. And that's important because many cancers very potently express CD24. So it's one of those, cell surface signals that certain cancers will express at very, very high levels, up to millions of copies per cell. And that has been used over the years as kind of a prognostic marker or ways to kind of subset different cell populations within a tumor. But now with this finding several years ago that Irv and Amira made, that this is talking directly to macrophages and suppressing them, we have a clear sense that it's a therapeutic target, because if we're able to disrupt this suppressive signal that it's sending to macrophages, then we'll be able to help activate macrophages to attack the cancer. So this is a signal that we think is especially important in a certain subset of cancers, mainly solid tumors, where we see this phenomenon of really, really high overexpression and or genetic amplification of the 6q21 locus where CD24 is present. So we think there's quite a handful of signals coming from the just observational tumor biology that this is something that the tumors have selected for. It's quite different from their normal tissue of origin in many cases. And then, now as we have started to look into developing drugs against it, we find that if you bind it and block it in the right way, you can get a really powerful macrophage activation that causes them to attack or phagocytose the cancer cells.
So armed with that knowledge, how did you develop or work on addressing that problem to come up with drugs to work on that?
It's a little bit of a different type of target, and I'll say a bit tricky from a drug discovery perspective. One of the interesting things that has been observed for CD24 over the years is that as it's been used as kind of a diagnostic or sort of academic tool to identify subsets of cells within the cancer, for example, some reagents have been developed against it, tool compounds like antibodies for immunohistochemistry. And interestingly, those different antibodies often show quite different patterns of staining on different cancers. And that, together with the knowledge of this glycosylation phenomenon on the CD24, has given us a sense that glycosylation, first, is important for the function of CD24, and second, that can be an important feature when thinking about developing antibodies. And so early in the company's history, even as we were building on the academic research that identified 24 generally as a target of interest, we started to understand that differential glycosylation would be an important consideration as we develop drugs against CD24. So a big part of our process was making sure that we were hitting the right versions of CD24 and antagonizing this pathway as broadly as possible in order to get maximum efficacy. And so that required us to, I would say, develop some specialized tools for our drug discovery pipeline. And ultimately, it was a really effective process. We've got a great protein science team within the company that developed a handful of different options against CD24. Amongst those different families, we see pretty different binding behavior depending on what type of cell you're looking at or what type of cancer you're looking at. And ultimately, we used a sort of empirical process to select the one that we thought hit the right balance between potency to induce macrophage attack on the cancers while sparing the normal cells as much as possible.
So how does Festo-1 work?
It's a monoclonal antibody against CD24. And we were looking for a drug that could have this sort of broad binding ability. As we kind of tested different antibody candidates against this target, we found, as between two different families, we might see equivalent binding on one cancer cell type, but then completely different patterns on another. And so we used that knowledge to find something that we found was minimally inhibited by different glycosylation patterns that might be present. In other words, it could bind to as many versions of 24 as possible. And we found that that corresponded very nicely with potency of immune activation. Having gone through that, we then sort of engineered the rest of the antibody from its original murine state now into a humanized antibody that compares very favorably against even fully human antibodies within the therapeutic landscape, and have now very thoroughly validated it for its immune activating activity. And we've come through manufacturing and are now right on the verge of starting our first clinical trial.
Is it something that is applicable to a whole range of cancers, or would you have to tweak it in order for it to be applicable to different cancers?
We think the drug itself is applicable to essentially all CD24 expressing cancers that we've encountered. But one of the biological things that we've found is that there's a certain threshold amount of CD24 that seems to be necessary in order to indicate, at least by our preclinical data, that it's a good candidate for an anti-CD24 therapy. If it's expressing the target to that threshold amount, we then see that Feast-01 is highly effective at inducing this macrophage activation. And so as we now prepare for the clinic, we're really interested to see if this set of preclinical observations that we've made, where in some cases we see a pretty clear correlation between CD24 expression and the efficacy of our drug, whereas in other indications, it's a less direct relationship. We'll see what we find when we get into patients, and we're interested to hopefully test pretty broadly, certainly including those patients who are going to be highly expressing of CD24 in those kind of disease categories that we see really have support across the board for this as a relevant target. And then, as we test our drug, I expect we'll also see patients that are a little bit more marginal for that expression. And we'll find whether selection for certain amount of 24 expression or, selection within certain indications is going to be a necessary part of our development pathway. I think it's the clinical data that will tell us that.
What did you find from your preclinical trials?
Well, we've developed a pretty large body of validation, both for the target and for the drug itself. In short, when you disrupt the CD24 signaling axis by deleting or blocking CD24, or, to a substantial degree by doing the same on kind of the other side of the pathway, blocking the inhibitory receptor on the surface of macrophages that can detect CD24, you see an increase in macrophage activation, and particularly in their direct effector function of phagocytosis, where they can surround and engulf and destroy cancer cells. So Feast-01 does that very, very effectively. As I mentioned, we optimized it for binding of all versions of CD24 that might be present on the surface of a cell, which in many cases will be heterogeneous even within a single cell. So we benchmarked this using some in vitro/ex vivo assays. One of our workhorse assays is to co-culture macrophages that we've derived directly from human donors, and then put those together with either cancer cell lines or, to an increasing degree, with primary tumors that we've fluorescently labeled. And then we can use either microscopy-based methods or flow cytometry to measure how much of the macrophage is eating. And so we see that in these key indications that are really our focus, where we're seeing in the databases like the TCGA, high-level expression of 24 across a majority of the patients, high frequency of genetic amplification of CD24. In general, then when you treat with Feast-01, you get a very potent activation of phagocytosis. That also carries forward into in vivo models. So we've validated Feast-01 in both the syngeneic setting, so in a wild-type mouse with a fully immunocompetent immune system, and in xenograft settings where we can directly test patient-derived xenografts or cancer cell lines that are really human in origin. And that's kind of quite specific to the macrophage function then in that system. We've seen both cases that Feast-01 is generally capable of driving tumor shrinkage, in many cases eliminating tumors and leading to long-term cure in those preclinical models. So we think that's an exciting set of data. Obviously, we've got a lot of data that makes the drug look great. And so we're using that as a way to kind of understand what is the ideal circumstance for using this in the clinic. We also have really pushed the systems to find circumstances where it's more difficult for Feast-01 to be active. And there, we think that's a good simulation of what the combination paradigms should look like in patients. Cases where, for example, they're chemo refractory, when we can combine Feast-01 together with standard of care drugs, we can sort of resensitize those to this combination therapy. And in general, one of the great things about macrophages is that by its mechanism of action, sort of anything that can make a cell sick or unhappy, even if it stops short of killing it, can also induce that cell to express certain stress signals, what we often call eat me signals, that then make it sensitive to macrophage attack, but only if you take away these barriers to macrophage activation. So altogether, I think we've developed a pretty clear sense that within this category of therapy, Feast-01 is incredibly potent. It's much more powerful, for example, than other macrophage checkpoints that have made it into the clinical stage, like the anti-CD47 antibodies. And we also find that it just plays really well with a broad variety of therapeutics that are already in use in some of these difficult to treat diseases where we're focused.
You mentioned getting ready for clinical trials. Where are you at with that, and what do they look like?
We're really excited about moving into the clinical phase. Obviously, we've been working toward it for quite some time. I think in the early part of this year, very soon, we expect to enroll our first patient and begin treating them. And that trial is, focused on establishing the monotherapy efficacy and safety of our drug. It's still quite a new target, and even though I think we've developed a pretty good sense of what it's likely to do and how it's going to interact with the immune system, of course, safety is the primary readout from this phase of the study. We also are keen to understand some of the patient characteristics that might correlate with response. And so in our initial phase 1A trial, it will be focused on a broad set of solid tumors, certainly including those that we think are the top of our list based on all these factors that I've discussed. But I expect also we'll have the chance to treat patients that are in some of these indications where maybe there's more heterogeneous expression of 24 between patients, or that might have, for example, high overexpression of 24, but not some of these other correlative signals like genetic amplification. As we go through 2025, we'll increase our experience in patients and start to get a sense of where it's going to work and perhaps what tox profile might be associated with its activity. And then from there, we'll be looking to expand into combination therapy, where I think for the immunotherapy space, that's often where we get the most robust responses.
How often do you have to administer it, and is it intravenous?
Yeah, it's a monoclonal antibody, so we're administering it intravenously. The frequency of administration in our first clinical trial will be every 3 weeks, which I think aligns pretty well with a lot of the other therapeutic paradigms that we might eventually combine with. But I would say that's an area that obviously we can understand what we see from the clinical data and adjust as time goes on.
You mentioned earlier about the fact that you're able to shrink tumors and maybe even get rid of them. What does success look like, I guess, for you in the long run? Is it curing people?
Our starting place is to provide a useful tool for physicians and to provide something that can derive real patient benefit. Like most therapeutics companies, and especially those in the immunotherapy space, our mechanism of action gives us hope that we might see really deep responses and perhaps quite durable responses. The indications that we're focused on to start, as you noted at the top, are difficult ones. And so I think there's quite a lot of room in between an inactive drug and a cure that would be really impactful for patients. And we hope to help provide that. Our key focus is that in these indications, there's just a clear lack of immunotherapy options. And that's something that obviously we've seen really transform the cancer landscape in those indications where they're available. And we hope to be able to provide that through our novel mechanism. Beyond that, even as we think to the long-term future, there's clear possibilities to combine with existing immunotherapies, or as I mentioned, these other therapeutic paradigms that are really firmly established in a lot of different diseases. And so I think the key will be to understand which patients could be relevant for our drug. And we'll start with those that are the highest likelihood of success. But we see a long-term path where if this works well in patients, then we might be able to, in perhaps a biomarker-informed way, expand far beyond those set of indications that are kind of at the top of our list. That's, of course, dreaming. We need to walk before we can run, but that's, of course, what we hope for patients. We're aiming to really provide what we hope to be a transformational therapy, a first-in-class therapy, and one that is clearly best against this target.
Clearly, there's a lot of work involved in what you're doing now, but is there anything else in the pipeline?
There is. I mean, even as we're focused on executing toward our clinical trial, since the beginning of the company's formation, we've had a really exciting discovery platform that we use to identify novel macrophage regulators that could potentially be either activating or inactivating. So that's a tool that might allow us to eventually expand even beyond our current focus in oncology. For now, I think we remain oncology focused. We just feel that there's a ton of untapped potential in this space. And so as we've used this discovery platform, we've identified quite a large handful of completely novel regulators of macrophage activity that have clear therapeutic potential. We've now gotten to the point of really prioritizing a specific target from among that class and are developing drugs to hit it in a couple of different ways. The one I think that we're most excited about that's likely to be our next program into the clinic is an ADC format that leverages this novel macrophage target to get immune activation while also delivering kind of a traditional ADC cytotoxic payload. We've seen in many different ways that these classes of therapies are just really powerful together and quite synergistic. And specifically, we find that by activating macrophages at the same time as delivering these ADCs, we get a dramatic sensitization of the tumor because we're bringing in this additional biology of immune activation. So we're really excited about the paradigm, both in a combination setting with agents that exist already. But as we look to the future, we want to develop a pipeline where sort of we can put all these pieces together in a single drug. And so we expect that to enter the clinic on the tail of Feast-01, but something that we have clear plans to advance into clinical development.
Very busy but exciting times.
It is. I think we've been really fortunate as a company. We've got, the benefit of starting from a really strong scientific foundation. We've been able to pull together an amazing scientific and broader team as well. And we've had great support from our investors. I just talked through the discovery platform. I would say not every company has the luxury of being able to pursue a clinical phase program while at the same time doing this basic science to, explore the fundamental regulators of macrophage activity. So I'm really grateful for that. And I think it's really given us a couple different paths to success.
Some great and very interesting work there from Feast. Let's hope their candidate turns that don't eat me signal into an all-you-can-eat buffet. Now to the other interview, and while you've probably heard of osteoporosis, you might not have heard of osteopenia, in spite of the fact that it's just as common. In fact, according to the Johns Hopkins Arthritis Center, 30% of Caucasian postmenopausal women in the US have osteoporosis, and 54% have osteopenia. To find out what it is, how it's treated, and how a new medical device is tackling osteopenia, we spoke with Osteoboost CEO Laura Yecies. All right, so before we get to looking into what you're doing, I wonder if we could get a bit of background on the company.
Sure. So Osteoboost Health is the creator of solutions, and our goal is to really optimize people's bone health through their lifespan. And very specifically, we are focused on the issue of low bone density, osteopenia right now, and osteoporosis in the future. And we have the first and only FDA-approved prescription medical device to treat low bone density. It is the only non-drug prescription treatment that's been approved by the FDA. And we are the first treatment that has really been designed specifically as an early intervention for patients with osteopenia, where our goal is that we can prevent their descent into osteoporosis. In addition to the medical device, we have a comprehensive solution for bone health and with a goal of preventing fractures. We recently acquired an osteoporosis exercise company called Wellin, and we are integrating that exercise program into our digital app, which goes with the belt and helps people not only with exercise, but with optimizing their use of the device, nutrition, and education.
You mentioned osteopenia. Could you maybe go into a bit more detail as to what that is and how it's currently treated?
Yeah, so good question, because a lot of people have heard of osteoporosis, and osteopenia, many people know about, but maybe not quite as universally. So, we start losing bone typically in our 30s, and that loss goes through a few stages. At first, you'll be losing bone, but it's still normal bone density. Then when you get to more than one standard deviation or a T-score of -1 below normal, then that's considered osteopenia. So you are at elevated risk of fracture, but not as severe of a risk as if you lose more bone and you're more than 2.5 standard deviations below average, that's considered osteoporosis. There's actually more patients with osteopenia than osteoporosis. In the US, we estimate about 52 million with osteopenia and another 12 million with osteoporosis. And low bone density is a common condition throughout the world. It's common in Europe, Asia, and those proportions, osteopenia, osteoporosis are similar. But this is really a global problem affecting billions.
How would somebody know that they have this?
That's such an important question, because before you've fractured, Osteoporosis or osteopenia is typically silent. You don't have pain from osteopenia. You can't sort of see anything visible. And so we call it a silent disease. And that's why it's so important to get screened. So the standard screening tool is a DEXA scan. It's a special type of X-ray. It's fairly inexpensive relative to other medical tests. Should be, maybe in the $100 to $200 range. Very safe, less radiation than a dental X-ray. And it really is a great tool to help identify what your bone density is and also to track it over time.
Is there anything that you can do to mitigate the onset of that as you're aging?
Yes. So, first of all, what I will say is most people will eventually have either osteopenia or osteoporosis. As we live longer, it's almost like high blood pressure. Most people will eventually get it. It's part of aging. But it makes a big difference if you lose your bone density early because then you'll continue to lose. So, what can you do to prevent it? So, first of all, nutrition is critical, especially, and of course, for maybe many of your listeners, it's too late for that, but especially in the teenage years, that's when the most bone is formed, but we continue to build bone in our 20s. But also after that, to mitigate loss, we need optimum nutrition. So that is, calcium, protein, and calcium for postmenopausal women. The target is about 1,200 milligrams. That's quite a lot for most people. So you really need to be conscious of it. Unless you live on a dairy farm, you're probably not naturally getting that much without some thought. And adequate protein, also things like, magnesium. Some people believe vitamin K and boron are helpful, but nutrition is important. Exercise, your bones respond to natural stimulation. And that stimulation ideally would be things like high impact exercise or really challenging weightlifting. Now, we do know that as people get older, many of them have difficulty doing that type of exercise, or even if they do it, they may not do it every day. So there's really, for most people, a gap in the stimulation that their bones are getting. Now, there's, many people who have conditions that lead to accelerated bone loss. So certain medications like corticosteroids. So, many people have been on steroids because of, serious asthma or autoimmune conditions. Those can really rob your bones. Now we're seeing that things like proton pump inhibitors for acid reflux. So there's a lot of things that can impact our bone health. And of course, genetics. So if you're like me and you're born with smaller bones, that's a risk factor.
Moving into your own company and what you do, I wonder if you could explain your treatment and how it was developed and how it works.
Sure. So the original research, vibration as a modality to improve bone health has been something that's been researched for a number of years. There's a body of evidence at a cellular level in animals, but NASA funded the first big initiative to research whole body vibration. And these are platforms that you stand on in astronauts. Astronauts, because of course, without gravity, their bones are not getting any stimulation. Astronauts lose bone density in space. In fact, the two astronauts that are stuck on the space station now, there's a lot of discussion about their bone health because over time it can really have an impact on them. So NASA studied using whole body vibration platforms to improve bone health. And there was some promising data, but the clinical results on the whole body vibration platforms have been kind of mixed. One of the things that we've observed is that, vibration dissipates as it goes up the skeleton. And so the results from those whole body vibration platforms tend to show bone density improvements in the feet, ankles, and lower leg. Well, of course, it's good to have stronger bones there, but the most common osteoporotic fracture is a vertebral compression fracture. The most serious, in terms of being the most deadly or debilitating osteoporosis fracture, are hip fractures. That's really where we are focusing right now to prevent those fractures. So that research was out there, but a bit mixed. So it showed promise, but none of those platforms were able to get an FDA Class 2 clearance, and they are not in wide use. So our inventor is a physical medicine and pain doctor, a physiatrist named Dr. Shane Mangrum, and he specializes in back pain. And he was seeing patients with these vertebral compression fractures, and unfortunately, there are no great treatments for them. And the more important thing is to prevent these fractures. Most of his patients expressed unwillingness or preference not to take the osteoporosis medications. And so it really stimulated his curiosity and passion to try to find a solution. So he came up with the idea of taking that vibration and applying it more locally to the vulnerable anatomy. So actually, the first idea was a vibrating chair, which kind of makes sense, right? You are closer to the hips and spine. But apparently, when they did some testing, that vibration was bothersome to many patients. It was vibrating their head, it made them feel nauseous or dizzy. And so the next idea, which turned out to be the winning one, was to put the vibration in sort of a wearable belt form factor. Now, that form factor gives you two important benefits. One is, and the most important one, is that the vibration is applied directly to the vulnerable anatomy. So the pack sits over the sacrum and applies the vibration to the hips and lumbar spine. And then as a wearable, it's just super easy to incorporate into daily use. We then went on to prototype this, and we were able to get an NIH grant to do a rigorous clinical study. And so we did a gold standard randomized control study at the University of Nebraska Medical Center. And that study was led by Dr. Laura Yecies. And what we saw was that with 1 year of use for patients who use the device 3 times a week or more, we evaluated their bone density and bone strength at baseline and at 12 months using CT and DEXA. And what we showed in the CT results was that we reduced the loss of bone strength by 83% and bone density by 85%. Results were also very favorable on the hip from the DEXA. And with those results, we applied to the FDA and we were granted a de novo clearance. I have to say that this is a pretty big deal for a startup. In the orthopedics world, the FDA has granted, I think, only 13 or 14 de novo clearances ever, and very few of them were granted to startups. So, this was a huge innovation, and I think really puts us in a very good position as a business. But most importantly, we have really for the first time a very novel approach for patients.
Does it work on different ages? Because obviously, as you age, your bone density decreases. Does the time at which you start the treatment make a difference?
What we know is what we studied, and then you can make some theoretical speculations. So what we studied was postmenopausal women with osteopenia. The age range was from early 50s to 70s. The average age in the study was 61. So we know it works for that group. We did see in this study that it worked. The effect size was a little bit bigger for the younger half of the study, the 50-year-olds. That is not surprising because our bones are more metabolically active when we're younger. And so perhaps that's the reason. There is some data that shows that exercise stimulus results in more bone gains when you're younger than when you're older. And essentially our mechanism of action is mimicking exercise from the bones. So, but exercise has been shown to improve bone density at every age. So starting early is great and encouraged, but it's never too late.
And is this something that could potentially be applied to other bone conditions? Is it something that you could maybe make something for
Yes,
ankles or?
right. I think there's every reason to think that a device optimized for other parts of the body would work. As I spoke about earlier, the whole body vibration was improving bone density in the ankles and lower leg. We've had many patients ask, could we do something for the wrists? Wrist fractures are pretty common. We're also very interested in seeing if we can develop something for more of the upper spine, the thoracic spine, which is also a fracture area. Although we did show efficacy at L1, which is adjacent to the first vertebra in the thoracic spine. So I think we are improving the lower thoracic, but we can't claim that. So yeah, I think we could certainly do other parts of the body. One of the things that we're trying to do is not just improve bone density, but prevent fractures. So how do you fracture? Have weak bones and fall. And so the osteoporosis exercise program that we acquired well in, it's our goal that that program can help people with their balance and general strength, and that that can reduce incidence of falls. We are analyzing that right now. And then we would like to investigate osteoporosis or low bone density that's not just, let's call the typical postmenopausal bone loss, which essentially our study was in healthy women, right? They had no other significant health conditions, just the fact that going through menopause and their age put them at risk of bone loss. And as I mentioned before, that's very common. But you have patients who, for instance, have more severe bone loss because of chemotherapy treatments, because of cancer that affects the bones, because of, as I mentioned, the drugs they need to take. And so we're very interested in seeing if use of our device could improve bone density in those patients who really need more options.
Is it something that people would need to use indefinitely, and how often do they need to use this?
I want to mention that the treatment is 30 minutes a day, so it's hopefully not too intrusive in the day, and you could wear it while you're doing other activities. Those results I quoted, the 83% reduction in bone loss, 85% reduction in bone loss, strength and density specifically, those were in patients who used it 3 times a week or more. So we definitely recommend that people aim to meet that minimum. But we did see in this study that patients who used it 4 times a week or 5 times a week had sort of proportionately better results. And so we recommend daily, or as close to daily as possible. And as I think your question implied, you sort of need to use this as long as you can, right, throughout your life. Just like, we need to eat healthy and exercise throughout our life. We need to stimulate our bones. And so this can help with that. But one thing that I want to point out is, let's say you used it for 5 years and then for some reason you didn't want to use it anymore. So our average patient had a 2.4% benefit to their bones in that year. So if you multiplied that out and then stopped using it, you're always that much better off for the rest of your life. And we also know that there is a kind of leverage in this model, a positive one. So, for every 1% improvement in bone density, you reduce fracture risk by much more than 1%. And depending on the study you look at, that multiplier could be 3% or 5% or even more. So even these small gains can be so impactful.
Are there factors that could both boost on the one side and diminish the effect on the other? Say, good nutrition and more exercise on the one hand, or poor diet, smoking, obesity, other kind of factors that would affect?
Oh, thank you for reminding me about the smoking, because when you asked me earlier, what causes bone loss, we do know that patients who smoke and people who drink more than 2 drinks a day, even with no other risk factors, on average lose bone faster and are at greater risk of fracture. There's a test that you can do, it's free and it's online, called FRAX, FRAX, to predict your fracture risk. And the FRAX asks you also about your smoking and drinking status and takes that into account, and it is significant. One of the things that we did in the study, which was quite important, was patients got calcium and vitamin D. Now, both the active and the sham group got calcium and vitamin D, but that is very important. And as I mentioned before, it can be difficult to get enough calcium. Many people, their vitamin D levels are below where they should be for optimum bone health. So, we encourage that for people using our device. And in fact, the companion app has a tracker to help you track your calcium consumption. And so we encourage people to do that.
When does it launch?
Yeah, we're looking at launching in the next month. So we're very excited about that. We did have a very exciting pre-order program. We went out to patients who were on our waiting list and gave them the opportunity to be first in line. So we see a lot of demand by patients. And the other thing we're seeing is, those patients need prescriptions. And we're having a number of doctors, prescribing. We've had a few endocrinologists prescribe more than 20 times, and doctors from more than 30 leading medical centers. So UC San Diego, Duke, Columbia, University of South Florida are prescribing. So we're seeing pretty strong acceptance from the clinical community. One of the magazines that goes to all endocrinologists did a 4-page article on Osteoboost, and I think that got the word out to the endocrinologists. We also had an abstract and a poster at the Endocrine Society meeting. And so what we're hearing from these endocrinologists is that they're excited finally to have a new treatment. Their patients want something that's drug-free, or maybe their patients need more in addition to the drugs that they're taking. And there are no new drugs in clinical trial right now. Most of the large pharma companies have abandoned the field. Merck, Pfizer, Lilly are not focusing on osteopenia or osteoporosis, despite the fact that there's so many patients. Amgen still has a strong program, as does Radius, but compared to cardiovascular conditions or diabetes, there's very little activity in the pharma area. And so I think there's a lot of excitement in the clinical community about Osteoboost.
How are you spreading the word ahead of its launch?
We're doing a fair amount of blogging and communication on social media, partnering with people in the field who are doing various explanations and analyses. We're going to these medical conferences. That's really a key place to meet with doctors. We do have a publication in the Journal of Bone and Mineral Research Plus. It's a very specialized journal, but the results of our study were published there. And so that's really an important way for us to get out to the physicians. It's interesting. Physicians who are focused on osteoporosis are beginning to be aware of us, but patients are actively searching for non-drug treatments and are coming to us. And what we're finding is they have been our ambassadors. So doctors have contacted us. I heard about this from my patient.
Does it have to be prescribed, or can people just buy it?
Yeah, it is by prescription only today. So, we want patients to work with their doctor. They should get their DEXA ordered, have a diagnosis, and the doctor can track them over time. We do have an e-prescribing solution for convenience for patients, but ideally they are being managed by their primary care or specialist physician to help really look at all the things that could be impacting their bone health.
And are there plans for this to be global at some point?
Absolutely, absolutely. We're hearing from patients and doctors all over the world. So we're a small company today. We aim to be bigger. So we've been focusing on the US, but we are actively talking to potential partners and regulatory bodies overseas.
It's always exciting when a new treatment comes out, whether it's a drug or a wearable device, because it means people suffering from a disease or a condition have more treatment options. So we wish Laura and Osteoboost lots of success with their upcoming launch. I know I'm going to make a fool of myself by announcing who is on the podcast next week, because the second that I do that, it will change. But as of right now, the plan is for our guests to be from Longeveron to talk about cell therapy and Alzheimer's. And if you've ever wondered what epiproteomics means, you'll have the chance to find out in an interview with Promise Bio. Even if you do know, please do tune in anyway to the new weekly Base-to-Base Biotech podcast. We look forward to hearing from you and to bringing you interviews with all the top biotech companies from around the world. So until next time, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next week for another Base to Base. The Base to Base Biotech podcast is an Air Coastal Media Limited production.